This is Nanoscape. I'm Seth Zimmerman.
And I'm Erin Spain.
On this show, we venture into the frontier of nanotechnology
with experts in the field who are turning the science of small into the next industrial revolution.
This podcast is brought to you by the International Institute for Nanotechnology at Northwestern University. Nanotechnology is helping a team of scientists and doctors here at Northwestern University tackle one of the world's deadliest cancers, glioblastoma.
Today, we're joined by Dr. Priya Kumthekar, a Northwestern Medicine neuro-oncologist who led a very promising early clinical trial in individuals with glioblastoma that showed an experimental spherical nucleic acid drug developed by Northwestern University scientists was able to penetrate the blood-brain barrier and trigger the death of tumor cells.
This drug, called NU-0129, is the first SNA drug developed for systemic use and opens the door to use this type of nanomedicine to advance therapeutic brain tumor care.
We welcome her to the show today to talk about the success of this trial and using nanotechnology in medicine. Welcome to the show.
Thank you so much for having me, Erin and Seth. Great to see you both again.
So right off the bat, tell us about your role at Northwestern and especially your work with these clinical trials.
Yeah, so at Northwestern, I'm a neuro-oncologist. So I see and treat brain tumor patients. So these are patients with primary brain tumors as well as secondary brain tumors. So tumors that start in the brain and tumors that actually come to the brain from other parts of the body and other cancers in the body. I also have a big role with clinical trials. So I'm the disease team leader for our neuro-oncology studies, which means basically that I oversee the portfolio of clinical trials that we have in the brain tumor space, again, in both of these categories, primary and secondary brain tumors.
Now, we talked about glioblastoma. This is a particularly very deadly form of brain cancer. Explain this to us and why no new drugs have been approved for glioblastoma in the past decade and why it's so critical that we do have some new drugs come on the market.
I'm so glad that we're giving this disease light because there's so much work to be done here. The disease glioblastoma, this most common primary malignant brain tumor for adults, is a really hard to treat tumor. So these are tumors that arise from the brain. They are in really any age adult, and they're difficult to treat for many reasons. So there's no current cure for glioblastoma. Probably one of the primary reasons why glioblastoma is difficult to treat is this entity called the blood-brain barrier, which I'm sure a lot of you have heard of. The blood-brain barrier is actually there for very good reasons. Our brain, I'm biased here, but it's the most important organ in our body, right? And so our bodies have developmentally created this really important barrier to keep toxins and bad stuff out of the brain. Well, as it turns out, therapeutics are considered toxins and bad stuff for the brain. So the brain has this natural physical barrier to keep those entities out. And so for that reason, we have a little bit of a hard time getting therapeutics to the brain. And it's not just the blood-brain barrier that causes difficulty in treating glioblastoma. It's also the disease itself. It's inherently really aggressive, and it's kind of a smart tumor. So there's differences in it, or we say heterogeneity in its tumor. So no two glioblastoma are actually exactly the same molecularly or biologically. Also, within the tumor itself, there can be differences. So we call these two things intertumoral heterogeneity. So no two tumors are exactly the same, and intratumoral heterogeneity, meaning you might treat one pocket of the tumor, but you might not be addressing 100% of it. So these are just kind of the tips of the iceberg in treating glioblastoma and the challenges that we see with it.
And then this is where nanotechnology comes into play. So we have a series of events that can only happen at Northwestern. Our own Chad Merkin, who we've had on a previous podcast, you should go back and listen to it. He introduced the world to spherical nucleic acids. This was in 1996. And this discovery has set the stage for what you were able to do in these clinical trials 2 decades later. Give us a little bit insight what these SNAs are and why are they such a good fit as a platform for glioblastoma drugs?
Yeah, so spherical nucleic acids, SNAs, are a fantastic fit, which I realized early on when I started to work with Dr. Merkin and others on this project, over a decade ago. But spherical nucleic acids, I think that they look like little koosh balls, because I know we don't have visuals here on this podcast. If everyone can imagine a little koosh ball with a core and then a lot of curly cues hanging off of it. I guess I'm aging myself with saying pushbox.
80s,
Everybody,
yes, very
our whole
90s.
audience is going to know what it is, but essentially a core. And for those of you guys who aren't children of the 80s or 70s like us, these are almost like curls that come off of a central core. And so what those are is genetic materials that can be tightly packed in a really small particle so that that particle can cross that blood-brain barrier, can sneak across that barrier that we talked about. And two, after it does so, it can have those little curly cues of genetic material start to make that biologic impact that we're chasing after.
So you mentioned this was more than a decade in the making, working with Dr. Merkin and others here at Northwestern from this initial concept in a lab that was carried out through preclinical research and an FDA approval and into a clinical trial. This is actually unheard of at Northwestern. This has never happened before. Can you tell me a little bit more about this journey?
Yes. And I think this journey is really so fascinating and highlights all of the key components and strengths that we have here at Northwestern. And Erin, before I get into it, I'll also say I'm really excited that this was a trailblazer for Northwestern. And you're exactly right that this hasn't happened before. But because of this project, this has happened afterwards. So this project was really a trailblazer. So I promise I'll get back to your original question now, which is what was that process? That process was basically that those preclinical studies that you mentioned. So we start in the lab setting, and to simplify things, we start with Petri dishes, and then we move to smaller animals. And the study that we did right before the human studies that we also published alongside the human studies within that Science and Translational Medicine paper was the non-human primate study, so the monkey studies. And so in these studies, what we gradually did was really understand the safety of this drug. We had gathered a lot of depth and understanding of the efficacy of this drug, so how this drug works, particularly in the glioblastoma setting. So what we did is then take all of the data that we gathered, right? Preclinical data, including our animal study data, and we designed a trial. But before executing on that trial, we met with the Food and Drug Administration to say, hey, how does this study look? What parameters do we need to carry this out successfully? So there's like these back and forths, and that's called a pre-IND meeting. So sorry for the jargon, but IND is an investigational new drug. And so whenever we do a clinical trial on a drug that's not approved in a certain setting, we have to obtain this from the FDA. There's a lot that goes into it. There's a lot of preclinical studies. We call them IND enabling studies, right? Because they enable us to get that IND. We have pre-IND meetings with the FDA where we say, hey, how does this look? Again, meetings that we do prior to getting that green light to say, hey, you can do your clinical trial. This is safe to do this clinical trial on patients, and the manner in which you're doing it is acceptable to us. So those are all the steps that it takes to actually get the trial going, not to mention making the drug. So in this process, we learned a lot about
only a small part of it.
Yeah. I'm sorry, I'm sound like alphabet soup here, but we also learned about a process called GMP, which is good manufacturing, production, and process. GMP grade drugs. We can't just have any drug made in a lab just given to patients. We need clinical grade that is good enough to give to our patients. There's a process and actually parameters that are set there too. So we had to go through all of these processes that took years and a lot of really smart folks to put this together. And then, of course, executing on the trial as the final step.
I have a follow-up question to that. The fact that this was a nanomedicine, did that come up at all in these back and forths with the FDA? And what role did the IIN and Dr. Merkin play in sort of explaining this to folks?
So absolutely it came up, right? Because this is the first time that this class of drugs has been put in the body for this disease. So it came up a ton. And so there was a lot of explanation. So the folks that you mentioned at the IIN, including Chad Merkin, were at the pre-IND meetings. They were discussing what do we need to do? What special precautions might we need to take? All of those things come up to ensure the utmost safety for our patients.
And then after all that legwork, you were able to recruit 8 patients to take part in a clinical trial. They were given the drug through intravenous infusion. And then what happens next? So what were the results from this?
So 8 patients was the maximum that we planned for. We planned for 6 to 8, but it was such a popular study with the patients that we accrued ahead of schedule and actually made the 8 patients, just as an aside, which is unusual actually for trials. So I just have to put that plug in. So the way that it worked is patients, as you mentioned, got the drug intravenously, then they underwent surgery, and then we analyzed their tissue. So what we found was essentially 2 major findings. Number 1, that the drug was safe. Actually, I'm going to add a third one, that this was safe and well tolerated. Now, these doses were a lot smaller because part of that FDA mandate and that back and forth with the IND that we talked about was that the FDA asked that these first studies be done with lower doses. So we did that. Nonetheless, we found, again, drug was safe. And then finding number 2, even with these lower doses, we found drug got to where we wanted it to go, which was the tumor. So that was our biggest feat, right? We got past the blood-brain barrier, drug got to where it needed to go. And that's really important. Arguably, the third thing is even more important, which is that the drug had its intended biologic effect. So what we looked at were essentially, to simplify things, we looked at markers in the tumor, and we looked at those markers showed and displayed the effects of the drug, NU0129. And we saw that, yes, indeed, it did show impact to the drug. And we compared that to the patient's original tumor tissue. So we had this nice kind of control group as well, as each patient served as their own control. So it was safe. It got to where it needed to go. And lastly, it did what we wanted it to do.
And again, just to clarify, I mean, this drug, it's not available yet to folks. It hasn't made an impact as far as curing this disease, but you've been able to prove something really important that hasn't been done before.
Yeah, absolutely. And I think it's really the implications for the future that those 3 things have that was really the major impact of this study.
And so now this SNA platform, you've shown that it can cross the blood-brain barrier. And this is a true game changer in a lot of ways. How could you see this nano drug, this drug platform being studied and used to treat other conditions, maybe outside of brain cancer?
Yeah, I think it can be used in many different scenarios. What we've shown here are multiple proofs of concept. We've shown 1, that we can get drug to where we need it to go. And there are a lot of brain conditions that we need to treat. It's not just brain tumors, right? There's everything from degenerative diseases, movement disorders, inflammatory, autoimmune, so many brain disorders where we're limited by this blood-brain barrier. And so we've really shown this proof of concept that with this technology, we can overcome that barrier in a safe fashion. I also think that we've shown the proof of concept of we can have that intended biologic effect with that genetic material around the core of our koosh ball, if you will. And I think the last one is kind of one that's probably near and dear to all of our hearts, which is it's really, my soapbox comment is it's very hard and unusual these days for novel therapeutics to come out of academic centers. And the proof of concept that we've really shown here is really kind of to cheerlead for Northwestern here, that we have all of the right ingredients to really take a smart idea from the lab and see it through all the way to our patients. And so that last proof of concept is one that we have seen since we've trailblazed with this project. And I think that we will just continue to see here at Northwestern.
So you published these findings nearly 4 years ago in Science Translational Medicine. So what was the reaction like, and what progress has been made since then?
The reaction was quite positive, amazingly positive for a study that just had 8 patients that was really aimed to be sort of a proof of concept study. We had phone calls from patients, physicians, truly all over the world wanting to know more or to be a part of it. So really, we were met with such an astounding positive feedback for this proof of concept that we showed.
So what has happened next? Has the drug moved on to a next phase?
Yeah, basically, the goal of the study was we used a gold core to make sure that we could trace it well. Now that we know it can go to the intended place, right now, we're looking at different iterations of that core and the halo around it. So we're basically iterating on the compound itself. And the next steps and the goals are to get those next generation compounds into our patients with glioblastoma in the future in clinical trials.
To be on the ground floor of such an important discovery in nanomedicine is really exciting. What do you think could happen in your lifetime in this field of nanomedicine? What do you see as the future?
Yeah. Well, I'll preface by saying I'm an optimist. I think most of us who are oncologists are in the field of in neuro-oncology, especially. So, I see a world where our patients have improved therapeutics to combat this terrible disease and really improve therapeutics to combat other neurologic diseases that cause strain in our patients' lives and their caregivers, and hopefully a world where maybe my kids become neuro-oncologists and they care for these very special patients and their families, and they have more weapons in their arsenal to help fight for these patients and give them the life they deserve.
What are some misconceptions maybe about clinical trials that you wish more people understood?
So for clinical trials, there's a lot of data that goes in prior to opening that study. That's not the first time that we're gathering data on whatever compound it is that we're studying, that we have the very least animal data, preclinical data, and we have some idea on its performance. At the same time, the opposite perception, I think I'd want to combat also, which is we know all the answers, right? Oftentimes I have patients who I'll present 2 trials to, and they'll say, well, okay, great, just tell me which one's better. And my answer to that is, gosh, I'm not holding back. If I knew one was scientifically superior, that's the one we would do. And it wouldn't be a trial, right? Because that would be an approved agent once we show that efficacy, especially in a disease like glioblastoma, where quite frankly, we get a lot of support from regulatory bodies to push forward drug development with things like orphan drug designation, breakthrough designations, and so forth. For our compounds, because it's such a hard to treat disease, the FDA is supportive of us trying to get drugs to patients faster. So I think that the 2 misconceptions are kind of on the extremes of we know nothing or we already know and we're doing this study. It's somewhere in that in between is where the truth lies.
I had a follow-up to something that you mentioned, that this was a first at Northwestern, but there has been a second now, maybe a third. Can you tell me about some of these other projects that you have literally been a trailblazer for?
Yeah, absolutely. So I'm really proud of a lot of the work that has gone on campus, particularly the past decade after this. So this was the first Northwestern-sponsored IND brain tumor project. And since then, we've had at least a couple more. So homegrown science, very much grassroots from our scientists here who have had great ideas. And instead of them sometimes falling by the wayside or going on to a different company, we've been able to bring those studies, those compounds to our patients here. And we know that we can do so now because we've developed that formula of how do we get that IND, right? How do we get that investigational new drug protocol? What are the steps we need to take to make drug? What are the steps we need to take to meet with the FDA? What are all those ingredients needed? We've baked this cake before, so we've been baking it a couple more times since then. And so it's wonderful to see.
So one thing that I read about you is that your research really balances advancing treatments while also prioritizing the patient's quality of life. So really, how do you approach that balance in trial design and patient care?
It's a tough balance. I mean, in trial design, what we really try to understand is patient-reported outcomes and quality of life. And thankfully, we have measures to do so, some of which have been created right here at Northwestern. So there's been some validated measures that we can look at to really understand how is this impacting a patient? Because our goal in doing these studies is not only just to have our patients live longer, but to live better. We are doing this so that they can live their lives. It's a conversation, right? Because some side effects can really impact a patient, and those same side effects might not impact another. So we definitely integrate it in 2 different ways, right? So in the clinic, day to day, we have these conversations just directly with patients and their families. How is this treatment impacting your life? What is stopping you from doing? Is it stopping you from doing anything? How do we balance that, right? Because sometimes the answer is, makes me a little bit tired, but I'm still able to do all the things I want to do. In which case, the patients want to keep going on whatever they're doing. It just it's very situationally dependent in the clinic setting. So having those tough conversations and making sure that we're bringing light to the side effects that their treatments might have. In a clinical trial setting, we do the same, of course, but we also capture data in a structured manner, capture things like quality of life endpoints, We want to capture things like patient-reported outcomes, and we have validated measures to capture these so that when we're reporting the results, we can say, well, this was the impact to the disease, but this is the impact to the patient who has the disease, which is, more important, right? Capturing all that data is really the answer.
This podcast is called Nanoscape, and we're really investigating nanotechnology in all these different areas and industries. And of course, we're talking about nanomedicine today. When you talk about nanomedicine to patients or colleagues or other just people that you know, what do you say about nanomedicine? How do you describe it?
Yeah. I say it's like medicine, but a lot smaller. No, I'm just kidding.
Yeah, I mean, perfect. Yeah, there we go.
Particularly with the study, which is the context in which I've used nanomedicine, we say exactly kind of what we've talked about today in a nutshell, right? I say, this is your disease. Here's the struggle that we have with therapeutics in our disease, right? So I'll mention the blood-brain barrier, and I'll say certain particle sizes can get past the blood-brain barrier. This is one of them because of its size. So we're using this therapeutic to get it to where we need to go, which is different than the drugs that we have now.
Thank you so much, Dr. Priya Kumthekar, for being on the show, talking about this really incredible clinical trial and the path that it has blazed for so many others. We appreciate it.
Yeah, congratulations. It's incredible. I can't wait to see where it goes next.
Awesome. Thank you guys so much for having me. This has been wonderful.
Thanks so much for listening. This podcast is brought to you by the International Institute for Nanotechnology at Northwestern University. Be sure to follow this show on Apple Podcasts or wherever you listen to podcasts, and be sure to give us a 5-star review.
For more information about IIN, go to our website, iinano.org/podcast.